Hi students, welcome back to the Chapter 5 lecture. You probably caught me mid-sentence in the last segment talking about how Lost River Pizza is probably not equivalent to a slice of Totino's pizza, for example. Anyway, that's all just to say that when we referred to calories in everyday language in regards to nutrition labels, we're actually talking about the kilocalorie. Okay, moving on to ATP and cellular work. So when we take in food, we need to convert the energy in those food molecules to ATP, which is the cellular currency that all of our cells use to perform their functions. So the chemical energy of organic molecules is released in the process of cellular respiration. And cellular respiration's goal really is to make ATP within these mitochondria, which are the powerhouse of eukaryotic cells. So what exactly is ATP? Well, it's a molecule known as adenosine triphosphate. It consists of adenosine plus a tail of 3 phosphate groups. ATP is broken down to ADP and accompanied by the release of energy. So if ATP stands for adenosine triphosphate, what do you think ADP stands for? I'll give you a moment to think about that. If you think adenosine diphosphate, you are correct. So what happens when ATP is broken down is one of those phosphate groups is released. And when you have adenosine plus 2 phosphate groups, you can think of it as adenosine diphosphate, which is ADP. And when that occurs, there's going to be a release of energy. So ATP can energize other molecules by transferring its phosphate groups. And this is the energy that can be used to drive cellular work. And this is all sorts of cellular work. So transport of large molecules into or out of the cell, signaling other cells, reception of hormones, all sorts of cellular activity requires energy. So, for example, imagine a bicyclist that's riding up a hill. In the bicyclist's leg muscle cells, ATP is going to be transferring phosphate groups to the motor proteins of those muscle cells, and the proteins are actually physically changing their shape as a result of this energy transfer, and that's going to cause the muscle cells to contract and perform the mechanical work of riding that bicycle uphill. So again, this is just a diagram of what occurs with energy release when adenosine triphosphate is converted to adenosine diphosphate. ATP is converted to ADP when a phosphate is transferred to another molecule. And there's going to be this release of energy which can be used to perform all different types of cellular work. One type of cellular work is mechanical work, so that example with the bicyclist, ADP is going to basically be driving this motor protein in a muscle cell to physically change shape and cause motion. There's transport work, so for example, if you have a large molecule, a solute that is within the cell and then in the extracellular space, you're going to need a phosphate group from ATP in order to make this transport protein that's spanning the phospholipid bilayer change shape and move one of those solute molecules into the cell. And then you have chemical work as well, so this can be used for all sorts of chemical reactions. And it's important to note that ADP can just be recycled back into ATP when a free phosphate group attaches back to ADP. ATP functions in what is called energy coupling, or the ATP cycle, which is just what I mentioned about ADP and a phosphate group being recycled to form ATP again. So whenever we expend energy, which is pretty much all the time, whether consciously or not, we're going to be spending ATP. Again, you can think of ATP as kind of like the currency of our cells. And ATP is recycled from the components ADP and phosphate through cellular respiration. So in this diagram here, we have cellular respiration being chemical energy harvested from fuel molecules. We're going to make ATP from that chemical energy. We're going to use ATP for cellular work, and then we're going to reuse those phosphate groups that were transferred to do certain types of cellular work. We're going to combine that phosphate with ADP to make ATP again. Okay, let's talk a bit about enzymes. Enzymes are just special proteins that assist in chemical reactions. And metabolism can be defined as the entirety of the chemical reactions that occur in organisms. So it's much more than just how fast you use food calories. It's the whole collection of chemical reactions that occur in your body. But from a non-biological science standpoint, a lot of people just use metabolism to say basically how fast their body uses calories. There are very few metabolic reactions that occur without the assistance of enzymes. And before we talk more about enzymes and their structure, I need to introduce this concept of activation energy. So this is the energy needed to cause a reaction to occur. And you can also think of it as the energy that activates the reactants. This is what triggers a chemical reaction. And you can think of activation energy as the amount of energy it would take to roll this boulder up this very steep hill. There's going to be an initial input of energy before you can get to point B in this diagram. So from a chemical reaction point of view, A would be before the chemical reaction occurs, and B would be after the chemical reaction occurs. So there has to be some input of energy here in order to get to those final products of the reaction. So without an enzyme, if you don't have this catalyst that's helping to lower the activation energy, you have this very high wall here that can be thought of as the activation energy barrier. There's just this barrier between getting from point A to point B. So there's a lot of initial energy that has to occur for this particular chemical reaction to take place. Now, if you have an enzyme, you can basically think of an enzyme as shortening or lowering this activation energy barrier. So it's going to make it much more energy efficient. You just don't need as much energy to get from point A to point B when you have an enzyme helping you out. So again, an enzyme is just a special type of protein. And once again, this kind of drives home the point that proteins are everywhere in our bodies, and they're basically vital for all sorts of chemical reactions and normal body functions to occur. So it's not just the protein-rich foods you eat, it's also all different structures in your body that are dependent on proteins, as well as all sorts of chemical reactions are dependent on these enzymes, which are also proteins. And each enzyme is very selective. You can think of an enzyme as having a very unique shape to it, and depending on the substrate that it's selecting for, it's going to have a complementary shape. So each enzyme only catalyzes one specific reaction. And when this substrate and this enzyme come together, you have what is known as an enzyme-substrate complex. Each enzyme recognizes a specific substrate, and the active site fits to the substrate, and the enzyme changes shape ever so slightly to get to that complex. This interaction is known as the induced fit. Another important thing to know about enzymes is that they can function over and over again. So it's not like when you have an enzyme, it just gets used for one reaction. It's basically a continuous cycle. This is a key characteristic of enzymes. So step 1 in this diagram, here you have an enzyme available with an empty active site. So this little unique shape here is known as the active site, and the enzyme in this example is sucrase. Another helpful hint here is enzymes that you'll see in this class end with ASE. So if it ends in ASE, it's an enzyme. And their names are often very closely related to the substrate that they fit with. So what do you think the enzyme sucrase fits with? If you said sucrose, you're absolutely correct. So sucrose is a common sugar, of course, and there's an enzyme called sucrase that will break down sucrose. Okay, so we have the enzyme here, sucrase. We have the substrate coming in that we've, for example, taken in with our food. We have some sucrose here. We're going to have the substrate binding to the enzyme. And again, that's called the enzyme-substrate complex when they fit together like that. The substrate is then going to be converted to usable products. And the products are released to be used in the body for energy. So we have fructose as well as glucose being the constituent parts that make up sucrose. Okay, I'm going to talk a little bit about membrane transport. So as I've mentioned before, there are proteins that will span the phospholipid bilayer of cells. And that bilayer is very important to keep the cell's internal environment in a constant healthy state. Working cells, they have to control the flow of materials into and out of that cell, and this is actually the primary function of the plasma membrane. Transport proteins also help with this task. So we have a wide variety of proteins here that are incorporated into that plasma membrane. We have, for example, a protein that helps with attachment to the cytoskeleton and extracellular matrix, so that would just be useful for cellular integrity and structural support. We have specific proteins for cell signaling. We have specific proteins for transport of large molecules that cannot flow freely across that lipid bilayer. We have proteins that help with intercellular joining. So if we have cells that need to maintain a close relationship for structural support or functionality, we might have intercellular joining proteins. Cell-cell recognition, which is particularly important for recognition of self versus others. And of course, we have proteins that are enzymes that fit right within that phospholipid bilayer as well. Okay, I'm going to go ahead and pause here, and I'll pick up where I left off in the next section.